So this final presentation of the morning is about what we call functional structural plant modeling. It is also modeling, and we also use it to understand intercrops and see, what is responsible for intercrop performance. But it's a very different type of modeling than the type of modeling that Sabine has just introduced to you. And it's also actually used for different types of questions. And I hope if there's one main take-home message from my presentation, then I hope it will be for you what type of questions in intercropping could potentially be addressed using functional structural plant modeling. If you feel that you are on top of that, then my mission is accomplished. Because then you have an extra tool in your gearbox if you're going to work on some intercropping question that could be useful next to experimentation, crop modeling, meteor analysis, and stuff like that. Yeah. So yeah, functional structural plant modeling. This is the type of modeling that explicitly simulates individual plants in 3D over time. Yeah, so in very short, that is what it does. So if you have experience with, video games, or sometimes you watch a movie with CGI created environments, you see all this lush environment, these plants growing, they are made using certain principles that we're also using in this type of modeling. Thing is, here we're using a biological fundament to make the plants develop and grow the way they are. And usually in these movies and games, that's not the case. It's purely for visualization. Now, I want to start with an example of an experimental observation that we did in an experiment back 10 years ago with, one of the earlier PhD students in our group that worked on intercrops with us, Junchi. And he was with us, and he was focusing on this interesting maize wheat intercrop, which indeed you do find a lot in China, in Gansu region, for example, where these are three stills, frames of the experiment. This is early on. You can see that, well, not so early on, but this is the earliest of the three pictures where you can see that the wheat is growing in strips of 6 rows, and the wheat plants are flowering. Right, and you can also see that the maize plants, they have just emerged not so long ago. So they were sown much later, obviously. In our environment, the C4 plant is sown later than the C3 species. So this is early on. This is around the time where the wheat has matured completely, and the maize plants have been overtaking the wheat in terms of height. So wheat plants have been harvested, and now in this stage, only the maize plants are left. And we did a couple of observations in this particular setup, and this is what people find in this particular intercrop, is that if you look at the yield in grams per meter of row, right? If you look in, we don't show it here, we also have the monoculture wheat, obviously, then you would get around 87 grams per meter in the sol wheat. And then if you look here, inner wheat, inner is the rows that are in the center of the wheat strip. Yeah, we get around 74, so that's a little bit lower, or I'm not showing any standard error here. So it's around, is more or less similar as the wheat plants do in a monoculture. But then if you look at the border wheat rows, yeah, so those are the wheat rows adjacent to the maize strips. Yeah, those wheat rows, they produce much more, triple. Yeah, so these rows are doing particularly fantastic. Well, I'm sure you get an idea why these wheat rows are doing so well, knowing what you know about intercrops, right? So why are they doing well? More light. They get more light. Why do they get more light? Because there's more space, right? Because these maize plants are still very small. Yeah, indeed. So, and that's above ground, below ground, same story, right? Water, nutrients, whatever. These maize plants are simply very young. Now, one of the reasons, one of the observations that we did on these wheat plants is that the plants in the middle, they actually have the phenotype of a normal wheat plant in a monoculture, but the plants at the border, they have many more tillers. Yeah, and this is a well-known response of tillering cereal plants and any grass basically to a low density or to low competition in general. These plants produce many more tillers. Why did you do that? Because they sense that there's space, that there are resources available to do that. So it capitalized on the situation that they're growing in. And apparently, in this particular intercrop, those plants, they sense that they know that they grow at a much better circumstances than the plants in the middle of the wheat strip. Now, this is what we call phenotypic plasticity, right? So plants adjust their phenotype to the local growing conditions that they encounter. And we had a question. This is still the same data. So here you can see the number of tillers per plant, like you see in the picture over time. It's not so interesting. There's another difference next to the number of tillers is the leaf size. This is interesting as well. I think if you go from the bottom to the top of the plant, you can see that leaf size increases. So the leaves get a little bit bigger as you go up the plant. But in the monocrop situation, am I saying no? In the inner row situation, at some point the leaves don't get bigger anymore once you go further up the stem. Whereas the plants that are in the border row, they still show an increase in leaf size. So the top leaves are bigger in the border rows than in the inner rows. And this is not just the case for the main stem, but also for the tillers. And knowing that these plants also have more tillers, so this multiplies, lots of leaf area on these border plants. This is also a plastic response. And then if you do measurements of light in these systems, this is what you find. This is the thermal time since sowing. So this is just the age of the canopy, basically. This is the fraction of PAR, so photosynthetically active radiation that you measure at a certain location in the canopy. You find that if you do that in the soil wheat, it follows a very well-known pattern. As the crop develops, the light at the bottom, I should say, so at soil level, goes down pretty rapidly, and most of the light is taken away. It's used by the plants for photosynthesis, right? If you do that measurement within the wheat strip, so in the middle of the wheat strip, you see the same pattern. There's a strange spike here. I don't know where this comes from, but overall, it's the same pattern. If you do that in between the wheat and the maize strip, so somewhere here, right, you can see that initially the light does go down, but then this drop sort of stalls. And then as soon as the maize plants take over, the light drops further. So we had a question. We know that the plants at the border intercept more light simply because they're at the border, but they're also responding to that plastically. And because of that, they intercept even more light. And we wanted to find out how much is this contribution of this plastic response of these wheat plants to the overall light capture by wheat in the intercrop. What I want to say is we wanted to disentangle the effect of being at the border from the plastic response that they show. So they get more light because they're at the border, but because they show plastic responses, they intercept even more light. How can we determine the contribution of these two? And we thought that's pretty difficult to do experimentally, right? So this is where modeling comes in. So these are typically the type of questions that we use this functional structural plant modeling for. It has elements of individual plants, plastic responses, 3D shape, and location in a heterogeneous environment. This is typically a scenario for functional structural plant modeling. So this is the example that I would like to highlight with you. But first, I want to shortly explain to you what is functional structural plant modeling. Yeah, so I dive a little bit into the past, don't worry, it will not be long. But I just want to show where this is coming from. There was this guy, Aristotle Lindenmayer, who was a biologist and a mathematician fascinated by plant shape and plant form. And he figured, these plants, they look complex, but actually they're not, because there's many aspects of shape and form in nature that look complex, but is in fact the same repeating pattern over and over. If you look at this fern leaf, for example, you can see this is the whole fern leaf. But then if you look at, you call it a leaflet, I don't even know. So this part of the fern leaf, it's basically a smaller copy of the whole thing. And then if you zoom in even further, this little piece here is also a smaller copy of the same thing. Yeah. So mathematically, this is called a fractal. Exactly. And you see these fractals popping up everywhere. in leaf venation patterns in this weird sort of broccoli type of, I don't even know what it's called. It's you know, it's a sorry. Yeah. Okay. Okay, Promanesco, whatever. It's a very nice 3D fractal. Here you can see these cones, and on these cones are cones, and then on those little cones are again little cones. Yeah, and of course these are biological entities. So if you zoom in at some point, it stops. Yeah, so it's not really a mathematical fractal, but still. And this is what Lindenmayer saw. He noticed that in plants you have this fractal pattern. Because if you just look at a very simple plant representation like this is the whole plant. If you look at the branch, it's basically a copy. And if you look at this sub-branch, again, it's basically a copy. So we realized if indeed these plants, we can consider them fractals, we can use very simple rules that if you keep applying them, gives this complex architecture. And this ultimately led to the birth of what we call L systems, Lindenmayer systems. Then there was this chap, whose name I'm not going to pronounce. He's from Canada. Well, originally from I think from Poland, if I'm not mistaken, but he lives already his entire life in Canada. And he teamed up with Lindenmayer. He saw, okay, and so this was a computer scientist, and he was interested in visualizing plants, and he noticed the L system from Lindenmayer, and he thought, okay, we can do something here. So he teamed up with him. Forming the lab, which he called Algorithmic Botany in Calgary. And together they wanted to produce lifelike simulations of plants. Yeah, so simulations that look like an actual plant. Now these are 3D simulations of, this is a lily here, and this is some, I forgot the species. And they created this in the early 90s. So these are pictures from the early 90s. And this all came together. It's very hard to read here. In this book, The Algorithmic Beauty of Plants, for anybody doing functional structural plant modeling, this is a must read, not because it's up to date, because it's not. It was actually published in 1990, 1 year after Lindenmayer passed away. But it is the history of this type of modeling, and it's really fascinating. Thing is, with these models, I'm going to skip this one. These models that show the plant grow and develop over time, biologically, they're empty. There's nothing in there. There's no biological concepts that give rise to the shape and the growth that you see. And for biologists, for these models to be interesting, they need to mimic plant behavior, not just the way plants look. Yeah. So there are really hollow shells regarding plant growth. And it was in the late 90s that people from biology, agronomy, they realized we can use these models to address questions that we have in living systems. Yeah, and this is one of the first examples of that, where such an L system-based model was made for maize by incorporating such biological mechanisms. Now, I won't go into the details of this one. I just want to go to the point where we are basically now. I mean, we're not in 2005, but in 2005, the foundation was laid for what we call right now functional structural plant modeling. So these are still the types of models that we use today. And this is also the type of model that I would like to show to you that we use to address certain questions in intercrops. Yeah, so the term was coined back almost 20 years ago and is still being used. So what are functional structural plant models? They're basically a combination of what I, a little bit derogatory perhaps, what I called hollow shells. They are, of course, beautiful mathematical, simple models of plant shape. But if you want to use them for biological purposes, they were combined with the process-based models that have been introduced also by Sabine in the previous presentation, that typically, now I'm cutting many corners, but that typically simulate plant growth and development based on uptake of water and nutrients from the soil and from light interception, and that contains these feedback loops. Because if your plants in your crop grow and develop, they can also, in the next time step, intercept more light and take up more water and nutrients. So these simulation models, we call process-based models, and in FSP models, so functional structural plant models, these two types of models are combined. And the earliest examples are from the mid-90s, specifically for trees. People in Finland were very much interested in simulating pine trees. And for cereal crops, now cereals are always a pet group of species for modelers. So it was adopted in the late 90s, and separately for root systems for people with a root bias in the mid 90s as well. So there were several developments here that ultimately came together. So the functional structural plant models that we use to address our questions in intercropping in our group, for example, but also in a couple of adjacent groups, they have a fairly similar setup. And in the following few slides, I just want to take you through a couple of mechanisms that are typically in such models to simulate the growth of plants in 3D and the capacity for them to interact with each other, to compete for resources. Yeah. So this is a simulated plant here, shoot and root, as you can see on the left. And when a plant grows a little bit, say the amount of growth that you get typically during a day, yeah, this changes the architecture of the shoot because all the organs, they grow a little bit, yeah, which means that the distribution of leaves in 3D space is changed also a little bit. Same goes for below ground. If your plant grows, your individual roots, they grow a little bit. And this changes the distribution of roots in the soil. Yeah, this is what plants do. Now, if the leaf 3D distribution in space changes, it also means that in this next day, your leaf light capture changes. There may be a little bit more light capture. If we're talking about a young plant, it may actually be a lot more. If we already talk about an established canopy, it may be only a little bit more because there's lots of self-shading going on. Yeah, but it changes the light capture. So together with the incoming radiation and the leaf 3D distribution, leaf light capture is determined. So this is light capture at the level of the individual leaf. Yeah, very detailed. This incoming radiation we normally simulate with a dome of light sources that simply cast their light rays onto the scene of plants that were growing there. And then this uses the principles of ray tracing. I don't know if you've heard of that. It's also, coming from the gaming industry, actually. Games, yeah, for gaming, but also to visualize stuff. Already in the 80s, people were using ray tracing to make computer images of glass balls on a checkerboard a pattern, for example. So it's a very old concept, but it's used a lot. And here we use ray tracing. So we're literally tracing rays from light sources towards the plant. Yeah. And then together with the optical properties of the leaf, so what do they do once a ray hits it? It is determined how much light is intercepted by each individual leaf. So that also means that the light intercepted by this leaf is different than by this leaf, simply because it's in a different position on the plant in the canopy. This captured light, together with the nitrogen content of the leaf, which is very important for photosynthesis, yeah, nitrogen content, and the prevailing temperature determines carbon assimilation of a particular leaf. Yeah, so photosynthesis rate. Then we go back to plant growth because the amount of carbon that's assimilated plus what we call sink strength, so the demand for carbon of a growing organ, that together determines how much an organ will ultimately grow. And this is calculated for each and every organ on the plant individually, which then feeds back on plant growth. Yeah, and this cycle continues with every simulation step. Yes. Yeah, that's a very good question. And this is, as Sabine said, for some of also for the crop models, and it's the same for these type of models. Some parameters in the model you can simply measure or derive from measurements. Some are very difficult to measure. So you need to assume something, you need to guesstimate them, right? But you can also calibrate them, which means you tune the output of the model by changing models that parameters you cannot easily measure. This is slippery thing to do because if you need to do that to multiple parameters, you may actually tune it such that it gets to your target, but yeah, you're right for the wrong reasons. So this is a difficult thing to do. But indeed, sink strength is a very difficult one to measure. I will get to how we measure this stuff a bit later. We're not done yet because there's also roots. And I would not dare to ignore the roots, of course.
I have a leaf bias.
Yeah, I have a leaf bias for sure. Yeah, that's why you are so complimentary. Yeah, so if we have a change in root distribution, this also means together with the amount of available nitrogen, in this case, in the different soil elements, this determines the nitrogen uptake by individual root segments. According to some rules, also sourcing related rules, the N is allocated to the different organs of the plant. So it ends up in the leaves as well, certain fraction, and this then helps determining, driving the carbon assimilation. Yeah, so this, for example, means that if you would grow this particular plant in a situation with low N in the soil compared to a situation with high N in the soil, there will be a feedback on how quickly this plant grows. Because there's simply not enough, and there may not be enough N in the soil to maximize photosynthetic capacity of the leaves. And if photosynthetic capacity of the leaves is lower, plant growth will be lower, which will also have an effect on the roots and on the shoots. Yeah, so these types of feedbacks are very typical for these FSP models. And this is also why you can simulate competition between plants. And that is the reason we use it for intercrop situations. Yeah, so to finish this scheme, I was talking all the time about growth of organs above and below ground, but not about the creation of new organs over time, which we capture in the term plant development. Yeah, now there's whole groups working on models for plant development, and that involves plant hormones and stuff like that. I'm not going to talk about that, also because for the purposes that we use this for in Intercrops, we don't normally need that. So plant development is simply a function of temperature. Yeah, if temperature is higher, plant development, so the production of new leaves and new side roots, for example, simply goes faster than when temperature is lower. So it's a very simple descriptive approach. You need to measure that, of course, and parameterize that, but it's a very simple approach. Yeah, I'm going to skip this. So what you end up with then, if you follow that scheme, is a simulation model that has the absolute bare bones basic biological processes responsible for plant growth in there. I mean, I didn't talk about water here. I didn't talk about phosphorus. Yeah, I mean, you can expand this indefinitely. I keep it to light and nitrogen for now. Which means we can, if you have the proper parameter values, you can simulate different species. For example, this is a model legume here, and this is a model cereal, with their very clearly distinct shoot architecture and root architecture. They are exactly the same models, just parameterized differently. Same principles. Now, obviously, if we have a module here for N fixation, then the parameter for N fixation in the cereal will be put to 0. Yeah, because there's simply some things that some species can do and others cannot. Now, what such a model allows you to do is already do very simple in silico, as we call it, experiments that are hard to do in real experiments. For example, see this intercropping situation. Yeah, we have a couple of cereal plants growing here and a couple of legume plants growing here. Same on the left and to the right. Suppose you have a research question about the relevance of root angle for the performance of plants in an intercrop. It's not so easy to do that in an experiment, right? You would need to have mutants somehow or varieties that differ in root angle, but then usually they also differ in other aspects. And mutants, if you knock out a certain gene, then usually this also has, you know, propagating effects on the rest of the plant growth. We see this in Arabidopsis a lot. So you can actually try to get a handle on what this may actually mean for plant performance in an intercrop by doing this sort of simulations. Yeah, and then the predictions may even be off if you just look at the pure numbers, but at least you can see in which direction your effect is going. So what, for example, what we tried to do here is we gave this cereal plants a much wider root angle than these cereal plants. And you can see that, right? I mean, these They nicely stay more or less in their own column. The plants over here don't. If you purely look at the cereal as a species here in this intercrop, and you look at the middle plants here, it's not going to make much of a difference, right? Because here, you know, roots from this side and this side are growing in, but roots from the plants above here are also growing outside of the zone. The net effect is more or less the same. But for the plants on the border, this is a different story, because these plants are now growing their roots in an area of the soil, a volume of the soil, where those legume plants are not putting their roots, or at least not yet. So that means that here we are using resources that we're not using here. So this was just a toy exercise, but the running hypothesis here was, okay, if we have a wider root angle, we actually get a better performance of the cereal plants, which may lead to a better performance of the intercrop as a whole. And then if you would elaborate this to a large research project, for example, you could imagine that this would be a tool to try and find traits that breeders could breed for in order to have well-functioning intercrops and efficiently functioning intercrops. Thing is, what we found here is indeed these plants, especially the ones in the border, they grew better because they had more access to resources. But what happened? Can you guess it? What happens if these plants have access to more nitrogen in this case? They will grow more. What happens if these plants grow more? They become
and more shading.
Higher, more shading. They become more strong competitors, right? So what happened here was that these legume plants were shaded a bit more by those cereal plants than in the left situation. And that reduced the legume performance by such a fraction that the total system performance effect was 0. Yeah. And to me, this to me highlights what is the potential use of these types of models. You can try and find out what is the pure effect of a trait on a single plant, but also on a community of plants, in this case, an intercrop, right? And you can test hypotheses very easily by seeing, okay, you know, does it impact my plant, but does it impact the neighboring plant? And what does this mean for total performance? How do these impacts stack up? Is one gain bigger than the loss, or is it the other way around? Do we have a net effect or not? So this is what we use these FSP models for. And now I want to get back to the example that I started with, because yeah, you've seen these pictures. We wanted to try and find out what is the contribution of the fact that these plants in the border rows produce more tillers with bigger leaves to total light capture of this intercropping system. We knew that this intercropping system was already over yielding, and we want to know what is the contribution of this plastic wheat responses to that over yielding by looking at the total amount of light captured by this intercrop. And we did that using such an FSP modeling approach. So this is what Junchi did in the past. He basically recreated these wheat maize canopies. You can see this. You can see it here in different stages of growth. And then what he did, and this is work also together with Wopke and a couple of other people in the group. Then what he did was exchange phenotypes. So what he did, and I'm just showing you a little bit of the canopy that he simulated, of course. He took the phenotype typical for a monoculture and the phenotype, so the number of tillers and leaf sizes, I call that phenotype now, typical for the different intercrop rows. And he said, well, assume now that I'm growing an intercrop, but the plants, they do not respond to being in an intercrop. They grow like they're in a monocrop. And by comparing these situations, you can say something. What is the contribution of showing these plastic responses to being in an intercrop to total light capture by the system? And this can then help us to explain where the increased light capture and therefore the increased performance of this intercrop system, where this is coming from, or at least what is the potential contribution of plant plasticity? That was the question we were interested in at the time. So that's what Junchi did. And let me just show you a couple of the results. We have a couple of simulation scenarios here on the y-axis. I will fill it up. And on the y-axis here is the accumulated light that has been absorbed in megajoules per square meter by maize and by wheat. So as you cannot see the gray, this is, okay, so it's a sol maize here, SM, sol maize, and this is sol wheat. Yeah, so this is the maize and this is the wheat on the left here. The dotted line, fortunately, you can see that one. This is the expected light absorption based on the land use of wheat and maize in this particular intercrop, which happened to be 1/3 sol wheat and 2/3 sol maize. Yeah, if we go back here, you can see 1/3 of the land area. Well, okay, this is not a good example. So if we disregard the left piece, yeah, then of this area, 1/3 is filled with maize and 2/3 is filled with sol wheat, because this is the repeating unit: maize, wheat, maize, wheat, maize, wheat. So this is the expected light capture if you would put the species together, if there is no additional effect of being in an intercrop. Yeah, you just take 1/3 of the sol wheat light interception, 2/3 of the sol maize light capture, and you add that up. If the intercrop does nothing to the light capture, this is what you would expect. Now, what did we find? So in all cases where there is a stack bar, the top is the maize and the bottom is the wheat. You cannot see it from the color. So this is what happens if you put sol wheat and sol maize phenotypes. So the plants, they look like they're growing in a monoculture, and you put them in an intercrop setup. You get a little bit more light capture. So apparently, by simply putting them in an intercrop situation, the fact that they are growing in that configuration already gains you a little bit of light capture. There's no plasticity involved here. Yeah, this is just having plants in an intercrop situation with plants intercepting light more from the sides in their strips and stuff like that. And then we introduced the intercrop phenotype, so the plastic responses to being in an intercrop. This here is the sol wheat with the intercrop maize phenotype. This here is the intercrop wheat and the sol maize phenotype. And then this is the full intercrop wheat phenotype and intercrop maize phenotype. Again, the top part of the bar is maize, the lower part of the bar is wheat. Now, if you look at the sol wheat plus intercrop maize, well, it actually goes down a little bit. If you look at intercrop wheat and sol maize, well, we get a lot extra light capture. This is, of course, also what we were expecting. If we have these tillering leafy wheat plants in there, especially in the border rows, you get a lot more light capture, and this is indeed what you see. With the sol maize still, if you also have the intercrop maize phenotype, it goes down again a little bit. Yeah, so this is what we expected, more light interception if you have this this intercrop wheat phenotype. What could be the reason, if you think about it, that the total phenotype, so also using the intercrop maize phenotype, the total light capture is again a little bit lower? What could that be? Yes.
Yeah, because the border rows are more competitive, and so they capture more lights, takes it from the other crop partner.
From the maize.
Yeah.
Yeah, exactly. Yeah, so these maize plants, they were having a hard time. Yeah, I mean, think about it. Consider you're a maize plant and you're popping out of the soil, and what you see to the left of you is a green wall of wheat plants. And then you look to the right a bit further, there's another green wall. You're having a really hard time. And in fact, I will not go into this now, but these maize plants, they also show plastic responses by extending their leaf sheets a lot, growing towards the light. So especially for our conditions, what is happening in this maize system is that these maize plants, they get such a shock and they cannot recover from it. So once the cereal has been harvested, you do get more light capture by the maize plant. Oh, sorry, once the wheat is harvested, you do get more light capture by the maize plants, because all of a sudden those pesky wheat plants are gone. And a lot of light is also hitting the maize plants from the side. But the season in the Netherlands was simply not long enough, and the temperature has been probably a little bit too low for these maize plants to fully recover from that and show the overcompensation that you sometimes see in other environmental conditions. Yeah, so here the maize plants were actually a little bit worse than in the monoculture, but the wheat plants did so much better than in the monoculture that the total system performance was much higher. Good luck trying to find that out in an experiment. It's very difficult. Yeah. And I also want to make a comparison to the crop models, the type of crop models that Sabine has been introducing you to. Those are perfect to do indeed surveys of or explorations of expected crop yield across a range of environments and soil types and plant types and species. Yeah, they are not made to make such phenotype switches, for example. Especially, I mean, now we did a full switch, either one or the other, but especially if you start to switch individual plants or perhaps even individual traits. Yeah, so there are simply particular questions that this type of modeling is useful for, and other types of questions where crop models are useful for. So pick your tool based on your question, I would say. Yeah. And this is the thing in a little bit more detail, just showing you how important this plasticity is. Now, if we just look at the wheat lines here, so disregard the ones on the right here, just the first ones. This is time, just day of the year, and this is the daily. Now it's not accumulated anymore, but daily light absorption by the wheat plants for wheat in the sole crop, which is the uninterrupted line here. Then the dashed dotted line, or whatever you call that, is for the sole wheat phenotype in the intercrop situation, so a little bit more light capture. And this line here, the dashed line, is for the intercrop phenotype wheat in an intercrop situation. And this difference is simply massive. So from this, we conclude that a lot of the extra performance that we see in such a wheat maize mixture can be at least partly attributed to the fact that those wheat plants respond plastically in the border rows, and we also put some numbers to it. Yeah, and this is something you can do with this type of modeling. Well, you can go further. I won't go into the details here. What we see is the different traits. So tillering, leaf size, leaf angle, and stuff like that. You can all turn it into the monocrop value or the intercrop value, and then test what is the contribution to light capture in a monocrop or an intercrop situation. And the take-home message of this chart is, wherever you see a high bar, there's a T involved. In other words, tillering. Yeah. So this, if it says T here, it means we use the intercrop tillering phenotype. And everywhere where there's a T, the bar is high, which means basically when you have the number of tillers that are specific to an intercrop, you're doing much better in terms of light capture. And the other traits that were tested, they had a marginal effect. It was really the tillering that did the trick. Yeah. Yeah. So we are using this type of modeling for questions also below ground these days. For example, also in the crop mix program, to which the experiment that we visited on Monday is part. We try to figure out how do plants compete for light, for water, for nitrogen, and for phosphorus. And then ultimately, we hope to be able to use this type of modeling together with the other things that we're doing in such a project, experimental work and meteor analysis work, to try and find out what type of plants do we need to have in order for, mixtures to function properly. So what type of management do we need to use in order to make that work? So in short, what do functional structural plant models do? They simulate plants interacting in 3 dimensions for resources. They are very well suited to capture the non-uniformity that is so typical for intercrops. For many intercrops, even the fully mixed intercrops, they are not as uniform as a monoculture, right? So they're really very well suited for that type of canopies. You can use them to quantify the relevance of particular plant traits for intercrop performance. We've seen the tillering example, but also the root angle example. Yeah, and they can be used to explore intercrop designs. This is something that I did not go into right now, but I'm sure you can imagine that if you can simulate canopies like this in the background, yeah, you can start playing with the number of rows in a strip, the width of a strip. You can play around with densities within the row. You can go more exotic and use totally different plant arrangements than using rows, right? I mean, in modeling, there's no limit, which is also the big pitfall of modeling. You can try everything. So if you don't start with a very clear question and hypothesis, you'll be doing months of modeling and end nowhere. And I've been there. And this is also the one thing that one of the things that I like to talk about a lot with the people that I supervise that are using this type of modeling. Please do not start, well, you can play around with the model a little bit, but don't start doing actual simulations before you have a very clear question and a very clear plan. What are you going to do with the model and how is it going to address your question? Otherwise, you're going to end up everywhere and nowhere. And you're not converging on anything. Yeah, so this is the pitfall of modeling, but it makes it a potentially very powerful tool for very specific questions. Last point I want to make is something that Wopke mentioned in the break when we were having a chat. Simplicity versus complexity. I'm very much I'm very much a proponent of trying to make things as simple as possible. So if things can be done simpler, do it simple. If you don't need complexity, don't use the complexity. And this is for me a very important point. If your question is on root angle, or perhaps the production of new leaves on a stem, or there's people that look at how leaves are bending, or what is the shape of a leaf, or how many leaflets does my tomato plant need to have. We've been doing projects like that. You need to have a tool that can capture this, but you need to have the simplest tool that can capture this. So even if these models may look complex because they have a lot of detail, they are not necessarily complex. They can be the simplest tool that you can make for that type of question. Yeah, if your question doesn't require a complex tool with a lot of detail like this, don't use it. Use this more simple approach. Right? So if you ever think, should I be using some modeling in the project that I'm in, or in the project that I want to go in, and you are wondering what type of tool to use, yeah, we're always available to have a chat about making a decision. And with that, I would like to end. Thank you very much.